I'm Barry Peake. I'm an Associate Professor of Environmental Chemistry at the University of Otago, and one of my interests is the trace metal content of a range of New Zealand organisms. Now, included in these are the New Zealand oyster, which is a very prized seafood around the world, and it's called the dredge or bluff oyster, and to give its proper name, botanical name, it's called Teostria chilensis. Now, this species grows on the seabed and is found at a variety of sites, at least around the South Island, but this is concentrated particularly in Foveaux Strait. Now, there's a small fishing fleet that goes out when the weather is appropriate and dredges in a season from March through to about August these oysters, brings them ashore where they are taken out of their shell and then sold to the consumers. Now, what is not known, of course, or obvious about these oysters is that they get their food from phytoplankton in the surrounding water. And the phytoplankton themselves absorb a range of trace metals from the seawater, incredibly small amounts, but it means that when the oysters filter the seawater, they take up the phytoplankton and in turn they bioconcentrate these trace metals. Now, they're present in the oysters in a very low concentration, which is generally doesn't provide any health issues at all to people who consume these oysters. However, their presence in the oyster generally depends on where the shellfish is collected. And so it provides scientists like myself the opportunity to characterize the area where an oyster has been grown and collected by carefully examining the trace metal content of the oysters from that site. Now, this also means that the origin of oysters purchased from, say, a fish shop such as this one can be unequivocally established by analysing the trace metal content of that oyster. And for government regulators such as the Ministry of Fisheries, this can have quite a useful benefit because it enables them to have an independent idea of where an oyster has been collected, and it could be that it's collected out of season. So what we do is take a sample of oysters, we shuck them, which means we take them out of the shell, we then weigh each one into a flask, and then we put it into an oven to dry overnight. And then we put it into what's called an ashing oven. And the purpose of this ashing oven is to burn off all of the organic material, leaving just an inorganic matrix. And then we dissolve up that very, very tiny amount of inorganic matrix in a dilute nitric acid, and then we run it on or analyse the trace elements in this solution in a very sophisticated instrument called an inductively coupled plasma mass spectrometer. Now, my research group at Otago over a number of years has undertaken these types of measurements on up to 8 samples of oysters collected from 14 sites scattered around the South Island, shown on this next slide, including 6 sites in Foveaux Strait and 1 out here on the Chatham Islands. Now, in all, a total of 111 oysters from 14 sites around the South Island were analysed, and here's a list of the concentrations expressed in units of micrograms of the metal per gram dry weight of the oyster tissue. So we've got to, because the oysters of course can vary in size, then the only proper way is to express it as a ratio of the weight of the oyster. The oyster of course also has variable moisture content, and so that's why we express it in terms of grams of dry weight. Now, this list here is clearly a big list, and so we can summarise all of this data by calculating the means and deviations of the concentrations for each site, as we've shown here in this table. This is a lot more concise summary of all the data, and we can see obvious trends, such as the cadmium levels here are elevated for the Foveaux Strait, whereas the zinc levels are elevated in other parts. But a better way of examining any trends is by a graphical means. And so the simplest way is to take the concentrations of each metal and plot them as a function of the site. So in this first slide, we have a plot of the concentrations of cadmium as a function of the collection site. Now here we've plotted the mean value of the cadmium concentration at each site with arrow bars representing 2 standard deviations of the mean. This gives approximate 95% confidence intervals. This sample size is small, and the exact confidence intervals would be slightly larger than the ones shown here, as they would be based on the t-distribution, which is not in your school's statistics syllabus. This plot further emphasises the higher levels of cadmium in Foveaux Strait oysters, as well as the Chatham Islands, and there is now a clear distinction between oysters collected at the other South Island sites. Now the corresponding plot for zinc is shown on the next slide, and apart from these 2 very high values, the trend is approximately exactly opposite to that of cadmium, namely very low in the Foveaux Strait and Chatham Islands, but higher in other parts of the South Island. So the challenge for a marine chemist like myself in looking at data like this is that if you're confident in the measurements that you've made, then you've got to think about what could be the reason for this trend. Now, an obvious possibility could be that at least the elevated levels of cadmium in Foveaux Strait might arise from runoff from farms in Southland that use a lot of fertiliser which does contain elevated levels of cadmium, and this would run off into the major rivers and into Foveaux Strait. But then if you stop and think a little more about this possible explanation, it certainly wouldn't account for the equally elevated levels that are observed for oysters sampled in the Chatham Islands. And so in fact, the explanation that we have for this phenomenon at present is that the phytoplankton that are present in the water and which the oysters feed on in Foveaux Strait and the Chatham Islands, the water is very low in zinc, and there is an active metalloenzyme containing zinc which is responsible for the metabolism of phytoplankton. Because cadmium and zinc have very similar chemical properties, then when the zinc levels are low, it is replaced by cadmium. And so the oysters feeding on these phytoplankton in turn pick up higher levels of cadmium compared to other parts where there isn't a zinc deficiency. Now, as we also note, there is a progressive decline in the cadmium levels in the Foveaux Strait sites. And so if we look in more detail at the map of Foveaux Strait, you will see that those trends and levels sort of correspond to a decrease as we go from north to south and closer to Stewart Island. But we really haven't been able to provide any further explanation. Now, returning to the zinc slide, you'll note this still leaves the question as to why there are these 2 very high levels for Bluff Harbour and Sawyers Bay, which is in Otago Harbour. Now, here I think the explanation is that both these sites, being close to relatively big urban populations of Dunedin and Invercargill, are going to receive stormwater, and the stormwater, of course, runs off roofs, rain off roofs, and the roofs, many of them have galvanised iron, which of course contains a lot of zinc. So we ascribe these elevated levels at these 2 sites close to urban centres as being due to runoff of stormwater, which is caused by rain falling on corrugated galvanised iron roofs, which of course contain a lot of zinc. Now, looking at other plots of metals of copper and manganese, there aren't any particularly obvious trends, or none that we can sort of ascribe to any particular phenomena. Now, the inverse relationship between zinc levels and cadmium can be even further or better illustrated by preparing a plot of zinc concentrations versus cadmium, as shown in this next slide. And it's very obvious here that when there's high zinc concentrations, there's low cadmium and converse. However, these scatter plots, if we prepare them for combinations of other elements, there's no obvious trends at all. Now, rather than compare the concentrations of just pairs of metals, it would be nice to be able to compare the concentrations of all 4 metals at once. And one way of doing this is to undertake what is called principal component analysis. And this type of analysis is one that you will learn at university. But essentially we develop two new variables which combine the effects of the concentrations of all four trace metals. The results of undertaking this principal component analysis are shown on the next slide, where we have plotted the means of the two principal components for the measurements of oysters from all 14 sites. And clearly now, and we've labelled these as groups, the sites from where they've come from, and you can clearly see that using this type of statistical analysis, there is a very clear distinction between the different sites, with a clustering particularly of Foveaux Strait and Chatham Island ones, which of course we know have elevated cadmium levels and low zinc in one part, and then we've got the Marlborough Sounds in another part, and then Sawyers Bay and Bluff Harbour. Now, we can use PCA plots like this as a guide to discovering the origin of an oyster by taking the oyster and analysing for at least these four trace metals, and then calculating the principal scores. We can plot it on this graph and then see if it fits in to one of these particular sites. Of course, if it was collected from somewhere else, it would fit in any particular part of the graph, but at least for the sites that we've chosen here, then we could confirm or otherwise that the oyster had been collected from that site. So we've now achieved one of the principal aims of this project, namely using statistics to process the raw chemical results, and then from this we can develop a very clear chemical signature for the origin of the oyster. And this clearly has implications for bodies such as the Ministry of Fisheries as they regulate the commercial oyster fishing industry in New Zealand. Now, we've also used this approach by my research group to look at trace metals in other New Zealand seafood, such as paua and scallops. We've also looked at fish, hoki and ling, and haven't quite got the same clear distinction as we have obtained with oysters, but certainly there are some good indications that we can identify where those seafoods are grown. I've also used it to identify the origin of cardboard and ginseng grown or manufactured or grown in New Zealand and overseas. So it is really a very powerful technique.